During interphase, the centromeres begin to move to opposite poles and the nuclear membrane begins to dissolve. During early prophase, the spindle fibers form and begin to pull chromosomes across to the opposite poles. During late prophase, these duplicate and and the nuclear membrane begins to constrict. During telophase, the nuclear membrane begins to rip apart while the chromosomes form inside of the nucleus. At the onset of meiosis, DNA strands thicken into chromosomes. Homologous chromosomes begin to approach each other. Homologous chromosomes pair to form bivalents. The centrioles divide and move to opposite poles of the cell. The bivalents duplicate to form tetrads, or chromatid groups. The nuclear membrane disintegrates and crossing over occurs. In metaphase 1, the tetrads attach to spindle fibers at their centromeres line up at mid-cell. In early anaphase 1, the tetrads separate and paired chromatids move along their spindles to the centrioles. In late anaphase, the chromatids have reached the spindle poles and the cell membrane begins to separate. In telophase 1, nuclear membranes enclose the separated chromatids, the cell membrane completes its constriction, and the first meiotic division ends. In prophase 2, the second meiotic division do not duplicate but merely separate. In metaphase 2, the chromatids line up at mid-cell and the centrioles and asters are at the poles. A spindle is formed. In anaphase 2, now separated chromatids approach their respective poles. The cell membrane begins to constrict, and in telophase 2, all 4 cells are completed.